Welcome everyone to the statistics seminars. And today is my greatest pleasure to introduce our speaker, Keegan Korthauer from UBC Statistics. And Keegan received her PhD in Wisconsin medicine, and then she did her postdoc at Harvard. And after her postdoc, she moved to UBC as an assistant professor of statistics. And today she's going to talk about very exciting topics on methods for sparse and high-dimensional measurements of DNA methylated.
That's great. Thank you so much for the kind introduction, and thanks for the invitation. I'm really excited to talk to you about my work here today at the seminar. Yeah, I'm not too far away physically, so it's a shame we have to do this virtually, but maybe sometime in the future I can actually come visit in person. That would be fun. So my work really lies at the intersection of statistics and genomics, and a lot of my recent work is focused on the analysis of DNA methylation sequencing data. And today, what I want to do, my main goal is to give you an overview of the types of statistical challenges that arise from this sparse and high-dimensional data. So to start off, we should go over a little bit of background because not all of us are necessarily experts in biology. So just as a quick overview, the study of epigenetics is dealing with the question of how, even though every cell in our body starts out with identical copies of DNA, we end up with many different cell types. And each of these has a unique and specific function that expresses genes at different levels. And one of the commonly studied mechanisms that explains at least some of this variation is DNA methylation. So what is this DNA methylation? So DNA methylation is just a chemical modification to an individual base on a DNA strand. And specifically, the modification occurs at the C base or the cytosine. And the modification in particular is when a methyl group, this M right here, is added to the C. This is referred to as this base being methylated. And when that mark is absent, as it is down here, then that C is unmethylated. And in mammals, like in humans, this almost exclusively occurs at Cs that are immediately followed by Gs or guanines. So when I refer to a site where methylation could or could not occur, I'll often call it a CpG. So that's just a C followed by a G. And in humans in particular, there's close to 30 million such available sites across the genome. So now we know what these marks are and potentially what they do functionally. How do we measure them in a sample? So how do we know where these marks are present or not present in a particular sample? So there's two main categories of methods that are in use today. The first on the left are called hybridization arrays. What these do is they use a predefined set of probes, and then they use fluorescence to assess the relative strength at each probe of methylated versus unmethylated DNA. And this is really a continuous signal coming from the arrays. And on the other hand, the other types of methods is sequencing assays. And these essentially use read counts to assess the level of methylation present. And this data is, like I mentioned, it's count data, so it's more discrete in nature. And so these two main categories of technologies, they have trade-offs in terms of the numbers of CpGs or sites that we can measure per sample, as well as how many samples can we analyze in a given experiment. So in this cartoon from a review study, the number of CpGs that you can measure per sample, which is referred to as the resolution, increases from left to right. And the number of samples that we can analyze per experiment, which I'm going to call sample throughput, increases from bottom to top. So arrays, which are in this area up here, so these are names of particular hybridization array assays, they tend to allow higher sample throughput. So you see they're further up on the y-axis due to their lower cost per sample. But the trade-off is that they have lower resolution. So they measure fewer sites per sample compared to methods that use sequencing. And then as well as sequencing, you get fewer samples because they are much more costly per sample. So with this trade-off, with this increasing resolution but decreasing sample throughput that you get when moving from the array technology to the sequencing technology, is this really worth it? So I want to talk about what do we gain with this increasing resolution, and also what are the challenges that it brings in the analysis. And in talking about this trade-off, it also helps to think about the types of questions that we want to answer. So with a low resolution analysis, let's consider two example CpGs or sites that are each measured in several normal and several cancer samples. And so here methylation level is shown on the vertical axis. And for each of these CpGs, we can see that they are differentially methylated. So they have different methylation levels in the cancer in red versus the blue normal samples. And if you did a simple t-test, you would confirm this. But do we really trust these signal measurements, or do we want to interpret them independently along with the close to 30 million other CpG sites that are present in the human genome? Well, if we move to a more high resolution analysis and in effect zoom out and see what the context is around these individual CpGs. So this is CpG1, again showing methylation level on the y-axis, but now showing a bunch of other neighboring sites along the x-axis. And the smooth line represents the average methylation level in each group. So here we can see that while the first CpG shows differential methylation still, the average methylation difference across the neighboring sites is close to 0. So that's in contrast if we zoom it out to where the CpG2 lies, we see a strong differential signal in the average methylation signal across the neighboring CpGs. So while both of them show this differential signal, it's often of interest to detect groups of CpGs or regions rather than individual CpGs. So for one, in some contexts, the regions may be the more relevant functional unit, and the CpGs are not acting independently of one another. And for this reason, a fundamental task in the analysis of this type of data and methylation sequencing data is to detect differentially methylated regions, which I'm going to abbreviate as DMRs. And this is enabled by higher resolution techniques such as sequencing, because if we may miss these if we only measure a small subset of these CpGs, like in hybridization arrays. However, as we mentioned with the trade-offs, the high resolution analysis means that generally we can only have fewer samples per experiment. And so we are left in this situation where we have high dimension, but really low sample throughput. In addition, this analysis task is not straightforward because detecting these DMRs requires us to account for site-wise correlation. So you can see in this example, there seems to be some spatial correlation of these methylation levels across different neighboring sites. And in addition, we don't, in advance, we don't know anything about the size or the location of these regions. So in the rest of the talk, I want to focus on these two main challenges of statistical analysis of DNA methylation sequencing data. So that's the first challenge is the limited throughput that limits the sample size that we have per experiment, and the complex signal that results from high resolution. And I'm going to go over a few examples from recent and ongoing work that illustrate the role of statistical methodology in addressing these challenges to ultimately, hopefully, extract epigenetic signal related to biological processes. So the first couple of examples are going to deal with what I call bulk sequencing. And this contrast between single-cell sequencing will become more clear when we get to that portion of the talk. And in the last portion, I'm going to talk about single-cell sequencing, which has similar challenges, but sparsity comes into play. So in this first example, we're going to discuss the task of inferring DMRs, so these differentially methylated regions, from a particular sequencing assay called whole genome bisulfite sequencing, or WGBS. This technique is something that can measure DNA methylation at extremely high resolution, so potentially every available site in the human genome. But remember, this comes at a cost that we can only measure a small number of samples per experiment. So this leads to analytical challenges. So first, to motivate specifically why we want to look for these differentially methylated regions. So a little bit more context biologically. So it's widely known that the methylation of CpG sites that are located in the region close to a transcription start site, which is near the start of a gene, this tends to be informative of the expression level of that gene. So specifically, as shown in this cartoon, where the promoter is not methylated, as shown at the top, the gene tends to be expressed or activated. And in contrast, when the promoter is methylated, the gene tends to be turned off or repressed. So this is just a general trend. However, this relationship has largely been observed through correlations of observational data, as shown in this scatter plot, where we have methylation difference on the X and expression difference on the Y. We see there are more points in this quadrant and this quadrant, meaning that there's this inverse relationship. However, despite the fact that we can observe these correlations, and sometimes they're quite strong across observational studies, it's still under debate whether the DNA methylation is a causal effect or whether it's actually a downstream byproduct of other regulatory mechanisms. So until recently, experiments could really only get at this question by looking at one gene at a time, which generally pointed toward causality, but they showed mixed results. And so a study from 2017 was able to ask this question for the first time in a genome-wide manner. So just instead of looking at a couple of genes at a time, look across the genome. And we don't have time to go into the details of the experimental procedure, but in brief, they were able to forcibly add these methylation marks to the promoters of thousands of genes and then measure what happens to the expression levels. And they did, based on the results, they came to the surprising conclusion that methylation was generally not sufficient for gene repression. So this potentially points at the methylation being a downstream byproduct of other mechanisms, not something that actually causes a change in gene expression. And this plot is from the paper by Ford and co-authors that summarizes their main findings. So here, each point represents the methylation increase and expression change in the X and Y respectively for a promoter region, where red points are those with an expression change that passed an FDR correction of 0.05. So how to interpret this is, we expect that if DNA methylation is causal for gene repression, that increases along the X should result in decreases along the Y. So we should see a strong negative trend. What we see, however, is that there's little correlation between the methylation and expression. And in particular, a histogram of the expression fold changes to summarize this for regions with at least a 30% methylation increase showed that almost as many genes were activated as repressed. So the results of this study, again, the overall conclusion was a bit surprising, and it's worth pointing out that this study used a computational tool that doesn't make full use of statistical inference. So in particular, the way in which significantly methylated promoters were detected and their increase was quantified does not control the false discovery rate, and nor is it able to account for critical sources of variability. So to understand this and the way the original analysis was done, we're going to go through this cartoon. So again, the original method does not take into account this critical factor of site-wise correlation. So we know that methylation levels of nearby sites tend to be similar to one another. They're not independent. So this original analysis used a method that essentially tests each CpG independently and then calls each one for significance or not significance. And then the ones that are deemed significant, if they are neighboring one another significant, then they're grouped together and forming a region. So that seems like a good place to start. But let's see what happens when we know the ground truth. So suppose we know that there's a true differentially methylated signal that we should detect in this location right here. But these other two sites are actually false positives. So when we examine the rate of false discoveries, which is just the expected proportion of the total discoveries that are false positives, at the CpG level, we see we'd have a value of 0.2, since 2 out of 10 of our total number of discoveries are false. So we might be fine with that. That might be the level we specified at the outset of the analysis, and that's great. The problem here is that if we look at that level at the level of DMRs, at the level of regions, we've made 1 out of 2 total discoveries, and our false discovery rate is 1/2. So we can see that what's happening here is that when we treat each individual CpG as a separate discovery, that's inflating the denominator of this calculation. And if we actually believe that these regions are acting as a functional unit rather than each CpG independently, this is going to be a problem. Since in practice, there's no way to estimate or control this level here. This is something that tends to be inflated. So in order to overcome this problem, and to be able to identify DMRs that are not contaminated by this inflated false positive rate, we developed a 2-stage approach that has 2 main features. So the first feature is that it controls for the site-wise correlation, and it does so using a permutation approach. So this approach, is important to note, works when we have very few samples. We have this sample throughput problem, so we could have sample size as low as 2 in each group. And in that case, we only have 2 unique permutations. And we want to permute the sample labels, but keep intact that correlation structure from sample to sample, or from site to site, sorry. And so we compare what we get with our actual data on this left panel and detect regions that look like they have a signal, and then evaluate their significance by looking at what is the signal that we get by chance when we permute the sample labels and keep that correlation intact. So this is going to control for that false discovery rate at the region level. And in addition, the way we construct the statistic, which we're not going to have time to go into, is borrowing information from across the genome and also further taking into account that correlation. So when we do, and this method is published, and there's an R package available in the bioconductor repository. So it's a general purpose method for analyzing and detecting these types of regions from this type of experimental data. And when we use this approach to reanalyze the data that was in the Ford study, we now see that using this approach, we see a strong relationship between the statistical significance of the methylation increase and the decrease in the expression level. So this is a figure that's analogous to the main figure in the Ford et al. Study, where again, if we think that this change is causal, then we should see a lot more points in the lower region of this plot, especially as you increase the methylation level statistic. And I'm highlighting this gene here, the SOX2 gene, as this was one of the main targets of the assay that they performed. So we expect a really strong signal in that gene in particular. And in addition, when we look at a histogram of all of the genes beyond a certain threshold, are they repressed or activated? Did they go up or down? We do see that the majority fall on the repressed side as expected. But what's perhaps more interesting is that with this approach, we actually can get an estimate of the significance at the region level. So this FDR, we can have an FDR cutoff of the DMR. So we can slide that over and say regions that have a significance, a higher significance threshold, and we can subject those to the same test and see that as we increase significance, statistical significance, we actually get an increase also in the biological signal. So even more proportion, a higher proportion of those genes are repressed until virtually all of them are repressed after a certain point. So this relationship, the fact that we see the statistical significance going hand in hand with the biological significance is a little bit of a validation that the signal that we're pulling out is relevant and explains a bit the differences in our results compared to the original study. All right, so now I want to move on to the next example, the second example. And in this example, I want to talk about another sequencing technology. And so instead of WGBS, the whole genome by sulfide sequencing, I'm going to talk about an example using a protocol, data from a protocol called MEDIP-seq. So MEDIP-seq is similar in that it's also sequencing, and it also looks at genome-wide methylation. But it differs in the fact that, well, WGBS gives us this relative signal of methylation and unmethylated. MEDIP actually measures only the methylation signal. So wherever we don't have a signal, we are left to infer that we don't have methylation, or it's just unseen. That piece of DNA is not seen. So it has slightly different challenges, but in overall, we have the same issue that the more we have the higher resolution, but we can have fewer samples than we could with array technology. And so we're going to talk about an example of using the signal and trying to detect a signal from this MEDIP-seq technology to predict disease status. And so to frame this prediction task, let's consider this plot, which shows a set of differentially methylated regions in pancreatic cancer samples compared to controls. So ultimately, we want to know whether we can use this set of DMRs, so this 14 set of almost 15,000 DMRs, and can we use that to detect the presence of pancreatic cancer in another cohort? Or is there a smaller set than this 14,000 that would be equally predictive? And note here, you can kind of see the rough sample size. This is not hundreds or thousands of patients. This is more on the order of tens of patients, because this is sequencing technology. It's not super inexpensive. So we have this challenge of low sample throughput. And in particular, we don't just want to know where these differences are. We don't want to stop at just identifying these DMRs. We want to know if we can find a set that's predictive of cancer status. Another thing to note is that in this particular example, the DMRs are identified from a particular type of biological sample that's called cfDNA, or cell-free DNA. So a quick aside on cell-free DNA. And what makes this particularly challenging is that this DNA, what we usually measure is DNA from intact cells. So we know we have a complete genome in our sample. Cell-free DNA is actually DNA that's leaked out from dead cells. And in a cancer patient, we expect some of this cell-free DNA will be actually coming from tumor cells, cancerous cells. But ultimately, the majority of cell-free DNA is leaked from normal cells that die of natural causes. So it's tricky in that we have these incomplete genomes, and we also have this mixture of tumor DNA as well as healthy normal DNA. And so we want to detect a signal that's ultimately, we're inferring comes from the tumor DNA, but we have this mixture. And this is a challenging signal. Even more challenging in this particular example, we want to be able to do this not only from blood plasma, which is a common source for cell-free DNA, but also in cell-free DNA from urine. So it's known that a limited amount of this cell-free DNA is shed in urine, but it's considerably less than found in blood plasma. So in this study, we focused specifically on kidney cancer, in particular renal cell carcinoma, with the motivation that we have a higher chance of detecting cell-free DNA in tumors in urine from a cancer of genitourinary origin. But ultimately, again, the signal-to-noise ratio here is going to be really low because of all these factors that I've mentioned. So it's really important to evaluate the robustness of our results, especially if we're not evaluating this in hundreds of thousands of patients. We just want to get an initial development to see whether there's potential to use this signal. So the statistical approach we used was designed to do this, to assess the robustness of these DMRs to predict disease status and try to make the most of our cohort of sample sizes on the order of tens rather than hundreds of thousands. And so we use a strategy that allows us to assess how sensitive the results are to which samples are included. So first, we randomly selected 80% of the sample as cases to train the model. And using those, identify the most predictive DMRs, differentially methylated regions, using regularized regression, in particular elastic net regression. And then evaluate the performance in a held out set of 20% of our original sample. And so the important part here is we're not going to just stop here. This is a very small cohort. It's a limited size. We don't have yet a validation sample to test it. And we really want to evaluate the robustness and whether it's worth it to try to do this on a validation set. So this process is repeated 100 times using Monte Carlo cross-validation to really let us see the variability in the development of this model. And so this gives us a measure. We can see the internal validity of the model building procedure. And so it's critical here to note that the feature selection of which DMRs are included is done inside the cross-validation procedure. So we're not selecting at the end of the day one final model. And as we can see here in this plot, we have the methylation score, so the prediction from our elastic net regression across different samples or in each box plot. And then within each box plot is the 100 Monte Carlo replications. So we see that there's some samples which are almost uniformly predicted to be in the cancer group. And we see that there's some, they're almost universally predicted to be in the control group, but then there's some in the middle that have quite a variability in the performance. And so depending on the composition of the training set, they get assigned quite different methylation scores. So it's really uncertain about those samples. And overall, we can average the performance across these 100 Monte Carlo cross-validations to get a sense of the robustness of them of these DMRs to predict disease status. That doesn't depend on one particular split of the training and test set in our data. So we can see from these results, it's encouraging that using the plasma that we see a signal. And do these have any functional biological meaning? So we can overlay these with DMRs we find from separate independent data, not from cell-free, but from actual tissue in a consortium cohort, the Cancer Genome Atlas. And we see that there's a weak but positive correlation between the DMRs that we identify as predictive and those that have differences between RCC and control in actual tissue samples. So there's some hint that what we're picking up on might be relevant biologically. But the challenging task here, we want to see, is this model procedure going to be successful using the urine? So the more challenging biological sample source. So we see similar results. We see there are some samples that are predicted with pretty high confidence to be in the cancer group and some in the control group, but overall the spread is much higher. And there's a lot more patients in this kind of middle range that have there's still a lot of uncertainty about their classification. And we see that also reflected in the average AUC and the individual Monte Carlo replication AUROC curves. And so overall, we expect, so this decreased performance relative to the plasma is expected as the signal in the urine is very sparse. Again, we don't have complete genomes and there's much less of this cell-free DNA than there is in plasma. And so there's a challenge of interpreting the absence of a peak as unmethylated versus just not present. And just to illustrate that, here's an example. This is from another cell-free meta-seq study, which really highlights the issue of sparsity. So this is a heat map showing regions that are differential between two different groups. And I just want to highlight a handful of samples here. You can see that there's very few regions with coverage in these handful of samples. And so the question here is, compared to the other samples, are these samples very lowly methylated? That's why they have a lot of white, or did they just have lower coverage and they had lower DNA material in their samples? So it's really something we can't determine without any extra information. And this problem is very widespread in the urine data. So this is why it's such a challenging prospect. And so just an aside and another iteration of this approach. So in collaboration with the same group out of Dana-Farber and Harvard, we tried to do another challenging problem, but from a slightly different direction. So moving back to plasma, but instead of trying to detect cancer versus healthy, can we detect two different cancer subtypes? So we know the patient has cancer, but we want to distinguish, do they have this more advanced and difficult to treat form of neuroendocrine prostate cancer, or do they have the more common and classical version of prostate cancer that responds more readily to treatment. So it's an important task to be able to predict these. But going into the study based on the results of our previous study, we knew we didn't want to take the exact same approach, that this problem was going to be more difficult than just detecting cancer versus normal. And we have this sparsity problem. So the approach that we ultimately took was to identify the features, do a feature selection based on an independent set of data. So it's specifically using a set of tumor tissue data, so not even using the cell-free yet, identifying our candidate features, removing some of those features that happen to be overlapping with white blood cell peaks. So we really want to narrow in the tumor. We don't want to get signal that's specific to blood cells. We want signal that's coming from this solid tumor. And then to get around the sparsity, took a creative approach to, instead of predict and use the elastic net to pull out from the candidate features which individual DMRs are most predictive, let's just sum across the entire set that we identified as potentially predictive and say what's the sum of the DMRs that are enriched in one cancer type divided by the sum of the enrichment in the other. So sort of a fold enrichment of the two competing signals, and then calculate that for each individual sample. So here we're trying to alleviate that sparsity effect. So it's okay if we have a bunch of regions that are not in every single sample, as long as you have some coverage across the set of hundreds of DMRs that are predictive. So if we do that on the test set, we see we get quite nice performance. And then further on a separate independent validation set, we see that still replicates. So we're quite encouraged by this type of signal of summing across the regions to deal with this problem of the sparsity. All right, so in the last 10, 15 minutes or so that I have, I'm going to move on to the last example, and we're going to move to another different type of methylation sequencing data. So we've kind of been down here in this chart so far, talking about these two sequencing approaches. And now I want to talk about sequencing of single cells. So single cell methods are actually not included in this graph that we've been bouncing around on. So this graph is from a 2010 review. So the single cell approaches were not in development yet. So I'm going to tell you that it's right in the neighborhood of this area. So this is because we have a huge increase compared to these other sequencing approaches in the number of samples per experiment, where we can sequence lots of cells, but we've moved a little bit to the left in the direction of the resolution. And this is because we're not going to get as many sites per cell, per sample, but we still do have base pair resolution. It's a little bit complicated about where exactly to put it. It doesn't really fall exactly along the x-axis on this graph, but it would be about here. It would move up in this direction. So single cell, the main difference is that up until now, you could think of a sample as a smoothie. So if you haven't heard this analogy, we essentially could observe the average of thousands to millions of cells. And you think of that as a smoothie, in contrast to single cell, where we can see in a measurement for every single individual cell. So that's more like seeing every single fruit piece in a bowl of fruit separately. And so this is exciting because we can start to see signals and attribute them to particular cell types, but it also comes with trade-offs because we can measure many, many cells in the experiment, but we typically observe far fewer sites than we would with non-single cell or bulk that we've been talking about so far. And so in particular, to give some concrete numbers as to sites that you have per cell. So in a typical experiment, the vast majority of sites we just don't have information on. And for the most part, this missingness is at random. So here's a cartoon of what a typical experiment would look like. So we have many, many sites. So this could be hundreds of thousands of samples of cells. And then we also have many, many sites. And this can be in a human around 30 million that we could have coverage for. But we have a lot of these question marks. These are missing values that we just don't observe. We don't know whether they're methylated or whether they're not methylated. And it's important to note that it's not the same set of sites that are missing in every sample. It's approximately, it's missing at random. There are some systematic differences we've identified, but essentially it's missing at random. And in experiments that we've looked at, it's typical to have approximately on the order of 1 million sites that you actually have measurements for in a given cell. So 1 million out of 30 million. So it's very, very sparse. And another difference here is you see for the observed data points, we have zeros and ones. So to a first approximation, we can think of this data as binary. And that's because when we look at just a single cell, we only have two copies. And so each DNA strand is either methylated or it's not methylated in a given cell. And the complication is that there is some small level of allelic variation. So your two copies of your chromosomes, they could have one allele is methylated and one not, but that is not the common case. The most common case is that they would be the same. And even if that is happening at an appreciable level, it's actually really hard to detect. You would need a lot higher coverage. So for simplicity, we just assume that these are binary. And the analysis task here, the thing that's interesting is similar to the task of identifying DMRs, but instead we want to detect variably methylated regions, or VMRs. So essentially groups of sites, groups of CpGs that show variable methylation. So maybe it's these three sites here that seem to have a different methylation level across our sets of cells. And these are informative these are what we want to use as input to, say, a clustering algorithm. So we can say something about, do we have some groupings, some subgroupings in our cells that are interesting? Do they have different cell types or different subpopulations, or are they in different states? So this methylation of these status of VMRs is informative of the subpopulations, as I said, but it's difficult to infer from this noisy, really sparse per cell measurements. But when we zoom out and look at the average methylation level across all cells, so averaging the binary values, we might see something like this. So if we have a VMR right here, we might see average level is, say, 0.7 across all of our cells. And what we don't see, but what we might think is happening, is that there are two subgroups of cells. One is present at 70% of our cells are methylated at that VMR, and the other 30% are not methylated. So we know that this, within a given site, this has to be the case, that there's only two groupings if we assume this binary yes or no methylation. And so this is just noting that the methylation tends to be correlated, and that we can find these regions where there's at most two groupings. So this is a key assumption. But it's really important to note that when we zoom out and consider different VMRs, it actually could be the case that we have some arbitrary number of subpopulations. So this is an example showing that maybe in total we have 3 different subpopulations. And in this particular VMR, 2 of those subpopulations are methylated and 1 is not. And that leaves us to the 50 plus 20% being 70%. But in truth, we could have any arbitrary number of subpopulations. And then when you zoom in far enough to these VMRs, the key thing is you just have at most these 2 groupings, again, stemming from the binary data. So this is work led by a PhD student in my group, Ning Shen, and she's developing an approach to detect these VMRs from the single cell methylation data. And this approach uses a hidden Markov model that determines the most likely number of groupings at any given site. So is there one grouping that's methylated? Is there one grouping that's unmethylated? Or are there 2 groupings, one that's methylated or not methylated? So these are the hidden binary states. And so VMR is just a consecutive grouping of sites that have the state where one is methylated and one is not. And to take into account the correlation that we know is present in this type of data, the transition probabilities from one state to the next are estimated from available data. And importantly, what we have as the observed data is actually aggregated over all of the cells. So aggregating the counts over the cells is what helps us to alleviate the sparsity and the fact that we have these random subsets of measurements that we see in each cell, and allows us to pool information across the cells. And the proportion of cells in each grouping, so in that previous cartoon example was 70% and 30%, this is allowed to vary by each region. So this model is fit for each region, and it's allowed to be different in every region, which is key to relaxing the assumption and allowing there to be an arbitrary number of total subpopulations. And this model can be solved by iteratively updating the states, the hidden states, with a Viterbi algorithm, and as well as the proportion parameters, which is, for example, the 70%, 30% with exponential gradient, and ultimately to maximize the likelihood. And this is just an overview of the workflow for the method. And essentially, it's similar to the DMR-seq framework. It's a two-stage. We first construct regions that look like they might have some variability. And then from those, limiting to those and detecting where in there we have these strings of regions that have the most likely state as being two groupings. And then mostly for computational complexity, we split up the computation to make it feasible and estimate on the longer regions and use information from the longer regions to inform through a prior the shorter regions, since there's not as much information about the shorter regions. And then ultimately the output is that we get these calls of where these VMRs are in the entire genome. And just some preliminary results to show that the modeling procedure performs comparable to or better than existing or more simplistic approaches. So here we used single cell bisulfite sequencing data from a 2017 study of mouse cortex cells, which were separated into 3 different hierarchies of cell types that were labeled from previously known cell type markers. So from broad, just 2-category hierarchy down to more specific granular 6 cell types. And in an unsupervised analysis, we evaluated the ability to recover these cell type labels using adjusted random index as the measure, and compared our approach VMR-seq with existing approach SC-MET, as well as 2 more simplistic versions of our approach. One which doesn't do the VMR calling at all, just uses the first stage of the candidate regions, just as a sanity check that we do better by being more selective than that. And then also approach that's similar to VMR-seq, but doesn't allow that proportion of subgroupings to vary. So it ultimately just assumes there's 2 subgroups. So you can see that those approaches don't do well, even though all approaches do relatively a good job on the broad, easy 2-category prediction. VMR-seq is the only one that does reasonably well on the fine-grained level 3 hierarchy. So just to conclude, since it's time to wrap up, so to conclude our tour of analysis of sequencing methylation data, I just want to briefly summarize the main themes regarding the statistical challenges in dealing with the sparse and high-dimensional nature. So I talked about 3 quite different examples, completely different data types, but they all had some feature in common, which was pooling information. And the motivation from that ranged from improving power to alleviating sparsity. So pooling information across sites, that was most helpful in the WGBS example. Pooling across regions ended up to be most helpful in the CF-MET-seq, and as well as pooling information across samples or cells was most helpful in the single cell BS-seq. So just to try to draw some similarities there. And another common theme that keeps popping up with this data type is adjusting for spatial correlation. So this is something we can't ignore. We know that it's a feature of the data, and we get into trouble if we try to treat these sites as independent. And we handled that in the DMR-seq approach by permuting sample labels to generate our null and keep that correlation structure intact. As well as in the VMR-seq approach for single cell, explicitly modeling the transition probabilities from data. All right, and then just a quick thank you to collaborators on the first 2 examples at DFCI, Dana-Farber Cancer Institute, and Harvard. as well as the Princess Margaret Cancer Center in Toronto. And then for the last example, the single cell, my PhD student Ming Shen, as well as I'd like to acknowledge that my funding sources from NSERC and BCCHR. And thank you to everyone for listening. And I wasn't interrupted with questions, so I hope you have some questions now for me. I'd be happy to take some questions.
Great talk, great talk. It's really nice. And okay, so Alan, I saw a question.
Yeah, that was a really great talk. I'm just curious on the last project, you used this comparison of RAND index to cell types. I've also done this kind of analysis before, and one question that I always kind of think of is like, to my knowledge, most of these cell types are called by other people clustering and then like looking at the RNA-seq and then deciding, okay, this is this cell type. So when you're doing this RAND index comparison, I guess what kind of is your justification for using whatever cell type the original people called to be kind of the ground truth?
Yeah, that's a really, really good question. And you really nailed the main challenge with that type of evaluation, is that it's not going to be perfect. And yes, it's results from earlier studies that determined that this particular cell type, if we're going to call it a cell type, tends to have the signal in this particular gene. And so we're going to look for that gene in the future studies and say whenever that's present, we're going to call that that particular cell type. We know that's problematic, and that and even if that was perfectly correct, that we could be very confident in those cell type labels, we're always going to be wondering about additional heterogeneity. So even in the most fine-grained cell type, it's not going to be the case that those cells in that particular cell type are necessarily going to be acting the same at all time points. Maybe there's some time of day effect. Maybe there's some microenvironment condition that those cells are responding differently in some way. So that is something that's missed by this type of evaluation. Ideally, you would have several independent sources of measurement to kind of pinpoint and narrow down these cell type differences. But from available data, that's the best that we can do at this point for getting some sort of ground truth. In addition, we're also evaluating with just pure simulation studies. But that's the best I can do. So that's still an ongoing challenge. Thanks for your question.
Yeah, thank you. And that's definitely not a knock on whatever you guys have done. You know, this is something that I do the same. You know, I was just curious what you thought.
Yeah,
Thank you. That was very helpful.
Okay, so next, Nick.
Hi, great talk. I guess maybe to try to connect this last project you talked about, the single cell bisulfate sequencing, with the one that was more about DNA methylation and like repressed expression. I'm wondering if would it be possible to do some kind of joint like single cell DNA and RNA-seq analysis? I don't know if in the lab you can actually like with one cell do both DNA and RNA-seq. I don't know, not a biologist, but it seems like it'd be an interesting way to connect kind of the functional variation to DNA methylation.
Sure. Yeah, so really, really great question. Yes, you actually can. There do exist laboratory techniques to measure both of those things at the same time in the same cells. Couple of caveats, they're both going to be very sparse. So you're not going to have a ton of overlap in the genes that you also have methylation for the promoters. So it's going to be one challenge. The second challenge is it's still not going to directly give you causality, because at the end of the day, it's still going to be observational data, unless you did something like the Ford and co-authors did, where you actually did a manipulation of the methylation. But otherwise, you would still just be looking at those two things at a snapshot in time. If that makes sense.
Thank you.
Great. And next one is Amy.
Thanks so much, Keegan. That was a really fantastic talk. And I really loved your emphasis on the out-of-study validation that you did recurring through all of your projects. So great work. I was curious about one thing that you said regarding differentiating between low methylation sites and sites that are sort of not present or not picked up by this technology. I think it was the second project you brought up. And I was wondering, do you have any like auxiliary information that you could use to distinguish between those, like, for example, like coverage or depth or number of reads for each of the samples that you have that might be able to allow you to distinguish those options?
Yeah, that's a great question. We've looked at a couple of things. Ultimately, I'm not comfortable using something like coverage or depth and asserting that is going to solve the problem. And the reason is, if there is just overall less methylation, you're going to see less total depth.
I see. Okay.
So there's still that issue that you don't have an independent measure that is met as some sort of control or reference that's out of sample. If you're comfortable saying that you don't expect a global difference in the signal, then something like the depth could rescue you and you could adjust for it.
Yeah, or even like marker genes. I don't know, do you have marker genes or marker methylated sites?
Yeah, that might be the. It's still, though, since we don't have complete genomes with the nature of the cell-free, the fact that you don't see the marker gene that you would definitely expect to be methylated, it could just be that you don't have it, or it could be aberrated. It could be an aberrant signal of cancer, and I could see which one you would be more suspect, but I wouldn't be confident calling. That said, there are developments by a group in Toronto, Princess Margaret, Daniel De Carvalho's group, that they're actually trying to develop, and I think have been somewhat successful at developing spike-in standards to really address this issue. And if you use a protocol that has that directly would give you that independent estimate of, how much signal do I have and am I recovering? So it actually has you spike in a known amount of this other DNA, and then that you should see it on the other end. And if you don't see it, if you don't see that signal, then you actually have some confidence of asserting that missing no coverage actually means that it's just missing material and not that it's lowly methylated. So you can try to tease that out.
Awesome. Great answer. Thanks so much. I had one other just sort of more technical question about, I think it was your first project. You had this really nifty video where you varied your FDR threshold and showed how varied the sites that you're looking at and showed how your FDR threshold, your FDR estimate varied. And I was wondering if, you brought up this issue between calculating FDR at the region versus the CPG level. And I was wondering, were the FDR numbers that you were looking at from the region or the CPG level?
Oh, great. Yeah, those were from the region level.
Oh, cool.
So I didn't really get into it because I wanted to talk about too many different things, but the statistic that it calculates it actually summarizes over all of the sites that are in a region. And so I didn't talk about the way it does that, but it essentially, you could think of it as looking at the average methylation difference across the region, and then comparing that with what you would see just by chance in the null.
Awesome. Thanks again for a great talk.
Thanks for your questions.
Great. Oh, Ellen?
Oh, sorry, I had another quick question that I was thinking about.
Sure.
So I know you mentioned kind of the number of samples in the single cell world. I was curious if you've given any thought to kind of how can you take into account both the number of cells, but also the fact that some of these cells come from maybe like the same biological samples. So like maybe it's like only 2 mice, but 30,000 cells. Is there anything that you've been working on or like stuff you've read about that like maybe tries to take that into account as well?
That's a great question. Yeah, there's certainly some additional heterogeneity. So we haven't directly tried to address that, but perhaps adding additional hierarchy in the model to account for that could be possible. Yeah, that's a great question. I haven't given it too much thought. In part because this data, it's a relatively new technology. There's not a lot of data out there that's available for us dry lab people to just start analyzing that we didn't generate ourselves. But I am excited that is going to be a problem very soon. We're going to have data on multiple individuals and multiple cells within those individuals to handle that heterogeneity.
Thanks.
Great. Any other questions? Okay, so if no other question, let's thank Keegan again for this very exciting and insightful talk.
Thanks, everyone.
Thank you.